Dear students of IPS 1209, Diversity and Classification of Organisms, which is a branch of biology known as taxonomy. I welcome you to this presentation. Like I told you, my left eye has been paining me, and the first time when I appeared in class at Makerere College School, I was actually feeling pain in the left and in the left eye, but I did not want to share it with you. And over the whole of yesterday and yesterday but one, I developed a hiccup. It is called Kasikonda, where I cannot breathe well, but the situation is being controlled. The situation is being controlled because I can now spend some hours without the hiccup, and I can now speak well. So I've been having two challenges, which is why I decided to record my lectures and share them with you. So I'm going to emphasize some concepts which you should also concentrate on as you're revising. Put your emphasis, your concentration on these concepts. In this video, you are going to get the notes very well and summarize those notes. You will be able to pass when you are assessed. We are going to keep the notes as simple as possible. Just elementary taxonomy, elementary classification, not complicated or advanced taxonomy. So I'm going to share my screen with you and kindly follow up what we are discussing. In the first slide here, which you can see on your screen, classification is the process of placing animals and plants into groups according to their similarities in structure, in physiology, and behavior. So when a taxonomist, a person who is specialized in classifying plants, plant taxonomist, an animal taxonomist, a scientist who is specialized in classifying animals, when a taxonomist uses the similarities in structure, the similarities in physiology, and the similarities in behavior of the organisms to place them into different groups, where each group contains shared features. All organisms in that group have got certain features or characteristics that they have in common, that they share in common. That is known as classifying or classification. So if a biologist, for example, puts all organisms which are able to move from one place to another place, and he places them in kingdom Animalia, then he places all organisms which are not able to locomote, which are not able to move from one place to another, and he places them in kingdom Plantae, where those organisms also share the ability to manufacture their own food by photosynthesis, the capacity for autotrophic nutrition. When a biologist places organisms with the shared characteristics in the same group, closely related members being placed in one group away from the distantly related members. Then that is called taxonomy. And remember in class, we said that this branch of biology known as taxonomy is divided into two main branches, where the first branch, we called it systematics. We called it systematics, which is about placing organisms in groups, both the living and extinct, both the living and extinct groups of animals, placing them in the same group. So diverse groups are formed, but on the basis of their shared characteristics. That is systematics. But if the scientist, if the biologist goes ahead to assign names, assign scientific names to each organism in that taxonomic group, and he assigns maybe a human a name of Homo sapiens. He assigns a housefly a name of Musica domestica or Periplaneta americana for cockroach. When a scientist gives names, scientific names to organisms, then that one is called nomenclature. And it is the second branch of taxonomy. So when we say the first branch of taxonomy is systematics, the second branch is nomenclature. Second branch is nomenclature, which is the system of naming living organisms, which we are going to see according to the rules. Each scientific name is assigned two Latin names or two Greek names. And the two Latin names, each of them must be underlined. If we are writing on paper, but if we are writing on a computer, we put each of those names in italics. But that assigning of scientific names to an organism is known as nomenclature. And it's another branch of taxonomy. Then the other one is systematics. So now, when you've collected the organisms and you observe their structural characteristics, and then you sort them into different groups depending on the shared features, each of those groups where you've placed those organisms is called a taxonomic grouping or a taxon. T-A-X-O-N, and the plural is taxa. So in classification, we create taxonomic groupings. So if you hear a taxonomic grouping of kingdom Animalia, all animals are in that taxonomic grouping called Animalia. If you hear another taxonomic grouping called kingdom Plantae, all plants, whether it is a tree, whether it is grass, whether it is a shrub, whether it is a herb, provided it is a plant, it will be placed in that taxonomic grouping known as kingdom Plantae. When we go to the mushrooms, the common bread mold, if we talk about that is rhizopus, the common bread mold. Those fungal organisms whose body is made up of those septa, and that are non-autotrophic. They cannot make their own food. They don't have chlorophyll. When somebody collects the mushrooms, the bread mold, or those the ringworms, for example, and places them in the same grouping called the fungi, that taxonomic grouping called the fungi is a separate kingdom. Is a separate kingdom. So in the taxonomy, in the science of classification, which we call taxonomy, we place organisms into recognizable groups, each group being called a taxon or a taxonomic grouping. But these groupings have a hierarchy, as we are about to see. The topmost hierarchy, in which all life forms are classified. The topmost taxonomic hierarchy, in which all living organisms are classified, is what we call a domain of life, a domain. So the kingdom is not the largest unit of classification in modern taxonomy, in modern classification. The largest unit of classification in modern taxonomy is known as a domain. So we can have a domain of eukaryotes, eukarya or eukaryota, that is composed of all organisms whose cells is made up of a true nucleus. And these organisms whose cells have a true nucleus are known as eukaryotes. And you will see that there are many kingdoms under that domain, eukaryota. You will find the plant kingdom there, because all plants have cells composed of a true nucleus. And what we are calling a true nucleus is one in which the genetic material DNA, which is normally a double helix structure, that DNA is enclosed by a nuclear membrane. That DNA is found in association with the protein called histones, to form a chromosome. So the DNA is a polynucleotide, a long molecule that is made up of units known as genes. And each gene determines the expression of a characteristic in an organism. So that DNA carrying all of those genes is found in a chromosome, a structure known as a chromosome. But all of these are bound or enclosed by a nuclear membrane that is perforated, that has small pores, tiny pores, which can allow exchange of materials between the nucleus and the cytoplasm. And we are saying that the genes carried by DNA or the chromosome in the nucleus are the determinant of the characteristics of an organism. The genes determine how the organism's characteristics will be expressed. Your eyes, your hair color, your skin color, your intelligence, IQ, your height, plant height, hemoglobin formation. Each of them has a gene in DNA in the nucleus that controls its expression. You've ever seen people called albinos, and some people are not albinos. It's because albinos lack a certain pigment in their skin for melanin production to give them a normal skin. So there is a gene in the DNA of albinos which is missing compared to the normal skin humans. The shape of your teeth, the shape of your nose, your eye color, everything you see on a living organism has a gene in the DNA, in the chromosome inside the nucleus, that controls its development and expression. And you've learned from cell biology that since DNA is a large molecule which cannot move out of the nucleus, it forms another smaller molecule known as a messenger RNA to carry the genetic instructions or the code from the nucleus to the cytoplasm, so that in the cytoplasm, during the process of protein synthesis on a ribosome, protein synthesis occurs on a ribosome in the cytoplasm. So on the ribosome, when this messenger RNA with the instructions from DNA attaches to that ribosome, those instructions, which we are calling the nucleotide sequence or base sequence or genes, will guide the cell as to which type of protein must be manufactured, must be constructed. If the body needs an enzyme, that particular enzyme will be the one to be constructed, to be developed on the ribosome, but using instructions that came from DNA. Remember, when DNA forms a messenger RNA, when the DNA exposes its part to form a messenger RNA, we call that one DNA transcription. So DNA transcription is the formation of a messenger RNA from DNA. And it takes place inside the nucleus. Then when we talk about DNA translation, we mean the building up of a polypeptide chain, the building up of a protein on the ribosome, using instructions on a messenger RNA, using information on messenger RNA that originated from the nucleus, that originated from DNA. So that is called DNA translation, or simply translation, the conversion of the information or instructions on the messenger RNA into a polypeptide chain or a protein. So we have all organisms in the domain eukaryota or eukarya having a true nucleus, one where the DNA remains in the nucleus, does not move out, but it controls all the cell's activities, including protein synthesis, enzyme synthesis, hemoglobin production, melanin production, fibrinogen production, cell membrane protein production, and all the proteins that you know in the body. both structural proteins and globular proteins. They are made using the genetic information in the DNA, and that is in all eukaryotic organisms or eukaryotes. And these are the plants, the animals, the fungi, and the protocysts, the single-celled organisms. Single-celled organisms, also known as protozoans, they form a whole phylum called the phylum Protozoa in kingdom Protocytista. So the 5 kingdoms under the domain of life known as Eukaryota or Eukarya is they include kingdom Animalia, kingdom Plantae, Plantae, kingdom Fungi, and kingdom Protocytista. That includes all single-celled organisms or unicellular organisms whose entire body is only made up of one cell. And they are some of the lowest forms of life that we know on Earth. Remember, when we go to the domain of life known as prokaryota, that domain of life, we have only the bacteria. And this bacteria, actually the true bacteria. But when you look at the bacterial cell, in the bacterial cell, we don't find a true nucleus, because after the cell wall of a bacterium, the next thing we see as we move inside is the cytoplasm. And within the cytoplasm, there is a circular DNA structure located in a special region known as a nucleoid. But that DNA is not enclosed by any nuclear membrane. Therefore, there is no true nucleus. It is simply a nuclear region, nuclear-like a region, but not a true nucleus. And these smallest organisms that are known to exist on Earth, the bacteria, evolved here on Earth thousands or even millions of years ago, before the higher forms of life appeared. So before plants, before animals, before the fungi, before the protocysts, the protozoans, the bacteria were there. And they have a simpler cellular structure. They are known as prokaryotes. I implore you to also read further, read more about the differences and similarities between the prokaryotes and eukaryotes. But one obvious similarity, one obvious difference is that prokaryotes lack a true nucleus, while eukaryotes have a true nucleus. And that means prokaryotes have a circular DNA in the cytoplasm, not enclosed by any nuclear membrane. Then the eukaryotes have a large double helix or double-stranded DNA that is enclosed in a nuclear membrane. And that is the most distinguishing feature between the prokaryotic organisms or prokaryotic cells and eukaryotic cells. The other difference could be the fact that the prokaryota includes only the bacteria, the true bacteria. While the domain eukaryota includes the plant kingdom, the animal kingdom, the fungi kingdom, and the protoctista kingdom. 4 kingdoms in one domain of life. So we've got very many other differences. For example, you'll find that eukaryotic organisms have a mitochondrion for cellular respiration or aerobic respiration. But when you come to prokaryotic organisms, the mitochondria is not there. And they may use simply a membrane known as a mesosome for carrying out the reactions of aerobic respiration. Respiration means the release of energy from food. Energy release from food. So there is a specialized cell organelle known as a mitochondrion in all eukaryotic organisms, where the processes of energy release take place within the cells, within the eukaryotic cell. But such a specialized structure, specialized organelle, is missing in prokaryotes. And you'll find that the means of reproduction in prokaryotes are simple. They are the simple means that we know, such as binary fusion and fission in general. And the processes of sexual reproduction may not be there. You find mainly asexual reproduction. But when we come to eukaryotic organisms, you'll find meiosis and mitosis, which are cell divisions or cell division types, take place. Cell division by mitosis and meiosis occurs, and there is also both asexual and sexual reproduction, where sexual reproduction takes place by formation, by production of gametes, by production of sex cells or reproductive cells known as gametes. But gamete formation or gametogenesis is absent in the prokaryotes. It's absent in the prokaryotes. And generally, like I've already told you, the bacteria, the prokaryota are simpler, actually the smallest cellular organisms. Yet the eukaryota includes simpler life forms and more advanced life forms. More advanced life forms. So get interested in prokaryotes and eukaryotes. You will find the bacterial cell wall is there present, made up of a material called peptidoglycan. But animal cells in eukaryotes, animal cells have no cell wall, but the cell wall may be found in plant cells. But the material in the plant cell wall is known as cellulose and semicelluloses. Cellulose and semicellulose. But that one in bacterial cell wall, I've said it is a peptidoglycan. So there are a lot of differences. Remind yourselves about differences between prokaryotes and eukaryotes. Why am I emphasizing this? Because the eukaryota and the prokaryota are the 2 main domains of life. So as I said, I decided to give you simple notes so that I don't complicate your life. We are going to cover the simple biology of classification that you know. And in these levels of classification, which we call taxonomic grouping, what you are going to add there as number 1 is what we've called the domain. The domain. That is the largest unit of classification. Not the kingdom, like it is indicated here. If I was in Microsoft Word, I would come here and add there the correct number 1, which is called domain here. So number 1 is domain. In that order, number 2 is kingdom. Number 3 is phylum. Number 4 is class. Number 5 is order. Number 6 is family. Number 7 is genus. And number 10 is species. So if they ask you, what is a domain? Say a domain is the largest unit of classification of living organisms, of living things. And you say a single domain, such as eukarya, contains more than one kingdom. It contains many kingdoms. Then if they tell you what is a kingdom, you say a kingdom is the second largest taxonomic grouping, and a kingdom is a subdivision of the domain. Kingdoms are subdivisions of domains. And within a kingdom are many phyla. There are many phyla, each of which contains distantly related organisms. In a single kingdom, we find many phyla, the plural of phyla, with each phylum containing closely related members, but the different phyla have got distantly related members, related organisms. Let me give you an example. In phylum Chordata, with the humans, we exist there. You find the fish, you find the reptiles, you find the mammals. But in phylum Arthropoda, you will find the insects, the worms, you will find the millipedes, the centipedes. In phylum Arthropoda, you find the insects, you find millipedes, the centipedes, the crustaceans, the arachnids like the ticks, the mites. So are we related in any way to the ticks or to the spiders that are in phylum Arthropoda? No. So you will find we are somehow related to the elephants, to the primates, to the carnivores in the same phylum of phylum Chordata. But when we go to another phylum, like a phylum Arthropoda, we are distantly related. We don't have any close similarities, any close links with them. And that means that within one kingdom, like a kingdom Animalia, you will find there very many phyla. But these phyla have members that are distantly related. But because they share some few similarities that are shown by all animals, then you will find all of them being placed in the same kingdom, Animalia. Also, when you go to the kingdom of plants, you'll find the trees, the grasses, the shrubs, the herbs, the mosses, the simpler ferns. They are all placed in kingdom plant. But look at a tree and look at a mosses that is growing on a damp wall on your house. Compare that mosses, the brownish organisms that do not even have a true stem, they don't have true leaves, they don't have true roots. but they have like root-like structures which are known as rhizoids. But the leaves are not there. But they are being placed in the same kingdom as the trees, such as mahogany. So meaning, what the taxonomists are interested in to place organisms in the same kingdom are just a few similarities. There are just a few similarities. For example, the possession of chlorophyll that enables them to photosynthesize, to carry out photosynthesis. So you'll find taxonomists putting the mosses, the tree, the grass, the shrub, the herb in the same plant kingdom plant. But when they are really distantly related, when those groups that have been placed in the same fire, in the same kingdom are distantly related. But when we move down the taxonomic hierarchy, similarities increase. Similarities increase. For example, all flowering plants will be placed in Angiospermatophyta. If they are placed in that phylum, Angiospermatophyta, because they have flowers, then you are seeing that the similarities are now, when we move down the taxonomic hierarchy, the similarities are now increasing. Meaning every plant placed in the phylum Angiospermatophyta will have a flower as a reproductive structure. Will have a flower as its reproductive organ. Then when we go to the coniferophyta, the conifers, you will find they also have their similarities. And the similarities are more compared to the similarities we find in the at the kingdom level. The similarities at this level of phylum are more. Now, when we walk down to the class, for example, among the flowering plants of the phylum Angiospermatophyta, you will find more similarities. What we will call class monocotyledony. Class monocotyledony. They will be flowering plants, but having their own features, such as leaves with the parallel venation. You will find a kind of fibrous root system, not a taproot system. So they will be the grasses. You will find them as grasses in their own class. The forbs in their own class called monocotyledony, but whose similarities are more at that level, at that taxonomic grouping, than at the level of phylum Angiospermatophyta. Then, when we go to another class, which is called dicotyledony, class dicotyledony, you will find the trees that have a taproot system, reticulate venation or network venation, and other similarities to cotyledonies in their seeds. You'll find all of them being placed in class dicotyledony with more similar characteristics compared to the phylum, compared to those shared at the phylum level. So as we move down the taxonomic hierarchy from domain to kingdom, similarities increase. When we move from kingdom to phylum, similarities increase. And now, going to the lowest taxonomic grouping, the species, we are going to find very closely related organisms that can even freely interbreed with one another to produce fertile offspring. At the level of a species, the similarities are more. They are more. At the level of a species, the similarities are more. The similarities among organisms in the same species are more than in any other groups, than in any other taxonomic groups above. So what you should note from that is that as we move down the taxonomic hierarchy, from the domain to the species, the number of similarities among organisms in that taxon, in that grouping, increase so much that the bottommost, the smallest taxonomic grouping known as species contains closely, very closely related organisms that are capable of interbreeding freely. They can mate with one another. They can freely interbreed to produce fertile offspring. So that is that. And please don't forget that order. And a simple memory or memorizer is here. That, but we can begin with the D. You can say dull kings. Dull kings play cards on fat green stools. Dull kings play cards on fat green stools, meaning that we are actually remembering with the first initial. The first initial dull is D, and that is the domain. K, it is for kingdom, P for phylum, C for class, O for order, F family, G genus, and S species, which is the smallest taxon, or the smallest taxonomic grouping. I'm not going to go into the details of what I've already said, but just to pass through this video and note these few things about each. I've said the kingdom is not the largest. Here you are going to try and say the kingdom. A kingdom is the second largest taxon after a domain of life. After a domain of life. And we have 5 kingdoms: kingdom Monera, kingdom Protoctista, kingdom Fungi, Plantae, and kingdom Animalia. Kingdom Animalia. Now, viruses are also known to exist on Earth, but they are not placed in any of these kingdoms because viruses are found at the boundary of living things and non-living things. At one point in the life cycle of a virus or a viral particle, it will be non-living. A virus is non-living. It is an inert organic particle when it is outside the cells of living organisms, when it is outside the cells of its host. If HIV is outside the cell of an infected host. It will be inert, it will be inactive, and the viral particle will be like a non-living thing. But if it finds its way into the host's cell, then it will begin showing characteristics of life. It will use its DNA or RNA to replicate within the host's cell. And what was a single viral particle multiplies into thousands or even millions of viruses when in the host's cell. When inside the host's cell. So I've already talked about all of this. Maybe what I need to emphasize here is that I said that taxonomy is divided into nomenclature, which is about naming organisms, and then systematics, which is about categorizing organisms into diverse groups, both extant, extinct, those organisms which have disappeared, and the present day living organisms. Now, nomenclature is the system of naming living things, the system of naming living organisms, was started by Carl Linnaeus. And the Linnaean system of naming organisms is followed. We follow that system, which Carolus Linnaeus, who was a botanist, termed binomial nomenclature, binomial naming of living things. And according to that system of naming living things, when you have fully classified a given organism, like a human there, from the level of a class, from the level of a domain eukaryota, kingdom Animalia, phylum Chordata, class Mammalia, order Primate, family Hominidae, genus Homo, and species Sapiens. When you have given the 8 taxa, the 8 taxonomic groupings, from domain here to the kingdom up to the species, then below here, you have to write and say, therefore, the scientific name of a human is Homo sapiens in italics. Or if you don't have a computer and you're using your pen, you underline Homo separately and sapiens separately, horizontally. Homo sapiens. Homo sapiens. I think we can even get a certain pen here. I was told that you can get a pen and you can highlight something. Okay, so that is the generic name, that is Homo. You see this PDF document, you can't easily edit it. Homo, and then this is sapiens. And what is common is that every name of genus begins with a capital letter. You can say Homo in humans. It is a generic name. Apis for the honeybee, that is a generic name. Z for the maize, that is a generic name. Agaricus for the mushroom, that is a generic name. Then when we come to the species name, sapiens, that is a species name, but it begins with a small letter. Now this is a mistake. It should be small m, mellifera for honeybee. Small m, maize, for the maize. Small c, campestris, for the mushroom. So that is what I want to emphasize, is that when we are writing a scientific name, please, we should go to we should go to the what? We should go to the generic and begin with. I'm succeeding to use tools. So you write it like that. Homo. With capital H. Homo. Like that. It's not easy to write using the tool. But it should be a straight line. Homo. Then you use a small s for sapien. Is on the same line, but you jump a small space, and then you write sapiens. Then after writing sapiens, you also underline separately. That is how we write the binomial names or the scientific names, which are usually in Latin, and they are italicized. You can italicize them instead of underlining if you are using a computer. Because if a computer, it just highlight, you'll just highlight, and then you go and put them in italics. So let us proceed. After binomial nomenclature, you will read these rules of binomial nomenclature, system of giving a scientific name to an organism, where each organism is assigned a scientific name with a generic name and a species name, underlined separately. The generic name beginning with a capital letter, and the species name beginning with a small letter. So this has already been explained to you. Humans are there, Homo sapiens, maize is maize, and you write the names. The names I shared them with you in class. Why do we classify organisms? It is easy to study organisms in a group since the members of a specific group resemble. Classification helps new organisms to be easily classified since they share certain characteristics with already those in existence. For easy identification, because organisms belonging to the same group show a lot of similarities, but also for easy communication and the prevention of confusion that is caused by the common names. So I will instead say Homo sapiens instead of saying a human, instead of saying omuntu, instead of saying omundu. In omundu is I think in Lusania or in Lugisu. So instead of using your Arabic name, Lugisu name, that brings a lot of confusion to the diverse group of people you are communicating to in the scientific community. Please restrict your language and your communication to scientific nomenclature, to scientific names. Okay. I want to end the first part of this video by discussing one of the kingdoms, which I said, these are the earliest organisms to evolve on Earth. They are known as the bacteria, and they form kingdom Monera. These bacteria are made up of one cell. They are made up of one cell, and we call them prokaryotes. But we distinguish bacteria from the single-celled organisms under Protoctista, because the single-celled organisms like amoeba, paramecium, and Eugerina, the trypanosomes, those ones, they have a true nucleus, and they also have membrane-bound organelles. They have cell organelles like the mitochondria. They have cell organelles like endoplasmic reticulum. They have cell organelles like the lysosome. They have cell organelles like the ribosomes. They have cell organelles like the Golgi body or Golgi complex. But they also have, the true nucleus, but those are absent in this structure of a bacterium. They are absent. This DNA which you see is naked. It is not enclosed by a true nucleus. It is not enclosed by a true nucleus. Yet if we are in an amoeba cell, we would have something like this, a true nucleus, where the nuclear membrane is enclosing a large double-stranded DNA molecule. And this double-stranded DNA molecule may be found in structures called chromosomes or chromatin. When the cell is in a non-dividing state, the DNA is in a form of, is found in a structure, in a body, a colored body actually. The word chroma means colored. In a colored body that we call a chromatin, that we call chromatin. So the chromatin in the animal cell, bacterial, amoeba cell, sorry, that is not an O, it is an N. It is an N, and it will read as chromatin. So DNA in eukaryotic cells is in the form of chromatin when the cell is non-dividing under a stage known as the interphase. But when the cell begins dividing by mitosis or meiosis, the name and the appearance of this genetic material changes, because the DNA is gonna be distributed from the parent cell into the daughter cells. So that DNA becomes what we call chromosome, not chromatin. But still, the word chromo or chroma is retained, it is maintained here, and it means colored. Meaning when you add there, certain dyes, this structure containing DNA can be easily visible in the microscope, and we call it a chromosome for a cell that has started dividing by meiosis or by mitosis. And the chromosomes will go through the 4 phases of cell division, beginning with the first phase called prophase. Next, the cells, the chromosomes of the cell will go to the second phase, which we call the metaphase. And then they will go to the third phase of cell division that we call the anaphase. Anaphase, just like you learned them in cell biology in semester 1. And then in the last phase, when the chromosomes are ready to separate and move into the daughter cells, we will have a phase called telophase. Telophase is the last phase of cell division. So in the in the stage of cell division, in the stages of cell division, the DNA will be in structures known as chromosomes. Remember, I said DNA is a polynucleotide, a very long molecule made up of nucleotides, and we call it a polynucleotide. But what it is carrying, the sequence of nucleotides in DNA are known as genes. And when I was beginning, I told you when I was beginning that there are thousands of genes in organisms. In humans, you find thousands of genes, but each gene has got a characteristic that it controls, that it determines. So all those thousands of genes, you will find them in a structure known as DNA that is found in a chromosome. So meaning that a chromosome is made up of two major components. The main component of the chromosome is a molecule which we call the hereditary material, or the genetic material of the cell, and this is known as DNA. Then DNA is associated with a protein known as histone. DNA is associated with a protein known as a histone, and the two form a chromosome. DNA associated with a histone forms a chromosome, or chromatin. When the cell is not undergoing cell division, we call it chromatin. So this is, like I've shown you on your screen, this is what happens in eukaryotic cells, that there will be a nuclear envelope surrounding that DNA. And this nuclear envelope is perforated. It has pores, tiny pores that allow exchange of substances between the nucleus and the surrounding cytoplasm. So the surrounding cytoplasm is the one which contains many cell organelles. And it is these cell organelles where the activities of the cell take place. So remember, the mitochondria are for aerobic respiration. The Golgi body is for secretions, like a production of mucus, production of hormones, production of secretions in the body. That is the Golgi body. All of those are found in the cytoplasm. They are cytoplasmic organelles. Now, this nucleus can also be looked at as a large organelle in eukaryotic cells. And we have already shown you that inside this nucleus, there is DNA, which carries the hereditary information, the genetic information of the cell. But being a large molecule, it cannot move out of the cell. It cannot move out of the nucleus, sorry. So what does it do? I said it forms smaller molecules known as messenger RNA that are single-stranded and small enough to move out of the nucleus. So the messenger RNA carries the information out of the nuclear pore, out of the nucleus, and it will come here and it will attach on a ribosome. And when it attaches on the ribosome, remember it is carrying instructions that are in form of the base sequence or nucleotide sequence obtained from DNA in the nucleus. And it is this sequence of bases or nucleotides being carried by messenger RNA, which will help in the process of protein synthesis. So during protein synthesis, one amino acid is selected using the instructions in messenger RNA, and it will be put in place. Then the second amino acid will be bonded to the first amino acid by a peptide bond. The third amino acid will also be brought to the growing polypeptide chain, and it will get bonded there via a peptide link. And the instructions for which type of amino acid should follow next are coming from the nucleotide sequence, from the nucleotide sequence of this messenger RNA, that line you see that is attached on the ribosome. This is the ribosome. That is the ribosome where protein synthesis takes place. That is the ribosome. Ribosome, where protein synthesis does what? Takes place. So we are saying the ribosome is only helping to attach the messenger RNA and to form a polypeptide chain that will form a primary protein, secondary protein, tertiary protein, and quaternary protein. All proteins that your body needs will be formed by this process. I said the first process where a messenger RNA is formed from DNA exposing part of its structure. That is called DNA transcription. Then this second process where the messenger RNA that has been formed from DNA moves out, attaches to the ribosome with its nucleotide sequence or instructions, and begins guiding the construction of a polypeptide chain that is called translation, amino acid translation. That is the information and the instructions in the messenger RNA are converted into a polypeptide chain and a protein molecule. So this is what takes place in eukaryotic cells, in cells of plants, in animal cells, in cells of protocysts, and in cells of fungi, in the fungal cells. But when we come to the bacteria here, which is the smallest cellular organisms, the protein synthesis takes place not on any structure. and you'll not find a lot of proteins being formed by that process. But the DNA which is present there will form some few proteins. That circular DNA will form some few proteins, and also it will help in the reproduction or replication, multiplication of bacteria by fission. By fission, that is that. So the main difference between the prokaryotic cells and the eukaryotic cells is that the prokaryotes or the bacteria have no true nucleus. Their DNA or genetic material is circular and small and found in the cytoplasm, not in the nucleus. But in eukaryotic cells, they have a large double-stranded DNA molecule that is confined in the nucleus. It cannot move out of the nucleus. It is exclusively found there. But it communicates with the surrounding cytoplasm through a messenger RNA. So that is called a true nucleus. The eukaryotes have membrane-bound organelles which are not found in the bacteria or in the prokaryotes. That is how we can distinguish the two domains of life. Important to notice that at this level, you should know how the shape of the bacterial cells helps in their classification. We can classify bacteria depending on the shape. Where the cells are spherical shaped, then we use the word coccus. So the name of the bacteria will end with coccus. If there are two pair, if there is one pair of spherical cells, we call that Diplococcus. If it is a bunch or group like this one, this one is Diplococcus, then this one is Staphylococcus. Staphylococcus. Then if it is a chain of spherical cells like that one, then the name becomes Streptococcus. Going to the rod-shaped bacteria that end with the word bacillus, or with the word bacillus. If it is a single rod, then that is bacillus. If it is a pair of rods, then that one is called diplobacilli. Diplobacilli. If the rods exist in a chain, several rods like this in a chain, then we shall call it Streptobacilli. So the names will be the same, but what changes is the suffix. Rod-shaped bacteria, the suffix is bacilli, Streptobacilli. Up there for spherical shaped cells, we called it Streptococcus. Diplobacilli for the rod-shaped bacteria. But up there in the spherical shaped bacteria, we have called it Diplococcus. We've called it Diplococcus. So like that, we use the shape of the bacterial cells to name them. Economic importance of bacteria. Read about them. Economic importance of bacteria. This, when we say economic importance, we mean both the uses and the harms or the disadvantages. For example, bacteria cause decay of dead plants and animals. Now that one is a positive importance, not a negative importance. Because when the dead bodies of plants and animals decay as a result of being fed on by the bacteria, then the nutrients which are locked up in their bodies are released back to the natural ecosystems to be reused by the living plants and animals, by the living organisms. So if we had a situation where there are no bacteria, then it means there would be no decomposers, there would be no decay, no rotting, and the dead bodies of plants and animals would be everywhere for thousands and thousands of years. But thanks to God for these decomposers like the bacteria, which can feed on the dead plants and animals, bring about decay, and release nutrients back for reuse in natural ecosystems. And this is called nutrient recycling. When we go to the nitrogen fixing bacteria, the nitrogen fixing bacteria, in the root nodules of leguminous plants, you will find this rhizobium bacteria, which is able to convert nitrogen from a form that is unusable by plants to a form which is usable by plants, to a form of nitrates that can be absorbed by plant roots. And when the plant roots absorb these nitrates, they will use the nitrogen to form plant proteins. They will use the nitrogen to form amino acids from which plant proteins are formed. They will use the nitrogen to form chlorophyll and other structures. This is also another good use. Bacteria manufacture vitamin B12 and vitamin K. This is capital K. Bacteria destroy harmful organisms in sewage. So this is an industrial application. Another industrial application is industrial processing of food, like yogurt, cheese, and vinegar. In the stomachs of ruminant animals, the bacteria provide an enzyme called cellulase. Cellulase, we simply change O to A. And that enzyme cellulase is used by the wood-eating termites, the wood-eating herbivores, the wood-eating organisms to digest cellulose, So without those bacteria that produce cellulase, cellulose digestion would be impossible. But this is a harmful, this is a disadvantage, though it is an economic importance. Bacteria could cause food poisoning and cause food to go bad. But also bacteria could deprive the soil of its nitrates. The denitrifying bacteria can convert the usable nitrates, the usable form of nitrogen known as nitrates, into free nitrogen, which escapes to the atmosphere, and that reduces soil nutrients, soil fertility. But we also know a number of bacterial diseases, in animals, wild animals and livestock, a number of bacterial diseases in humans. And pharmaceutical industries manufacture antibiotics simply to help us kill these bacteria and to treat the bacterial diseases. So, ladies and gentlemen, the first video is actually about the introduction to the science of classification, which we call taxonomy. And like you will see in these videos, the second component of this course, which is called diversity of organisms, which we also call biodiversity or biological diversity. That second component of biological diversity is implied in classification. That as we are classifying these organisms, you will be surprised that they are very diverse, very diverse. Very diverse meaning there are many species in one genus. There are many genera in one family. There are many families in one order. There are many orders in one class. There are many classes in one phylum. There are many phyla in one kingdom. And in one domain of life, which we call eukaryota or eukarya, there are 4 kingdoms. So that is how diverse these living organisms are as we are classifying them. But we only try to bite something small which we can easily chew and swallow. Otherwise, this branch of biology that deals with the classification of living things is very broad. Taxonomy, which deals with the classification of living things, is a very broad branch of biology. So I implore you to go and review this video. Go and review this video. Note down as you are reading those notes and as you are listening to me, note down. There are some things which may not be on that study material which you see in the video. But as I am conversing through the study material, there is some information that you get. There is some information, extra information, which builds you into a good biological scientist. I wish to stop here in the first video, and I request you to share them across the entire group using your WhatsApp. Use your WhatsApp groups to share my videos. Use emails to share my videos. Let these few remaining weeks be weeks of individual revision and even group revision, writing down and summarizing these ideas. You will pass when you are assessed. Remember, we will have to do a test. I want to give any coursework, I'll give a test. So you have to do a test and also an examination. Whether it is a test or exam, section A is composed of 15 multiple choice questions. Section B is composed of 10 short answer questions. And section C is composed of 5 essay questions, of which you can select only 3. And you will be done. The semester will be done. But I don't want you to be put on exam pressure. Just enjoy the branch of biology known as taxonomy by revisiting these instruction resources. Thank you very much. I wish you the best. So I'm going to send you 2 videos at the same time. One video you'll be able to see me, my face, like you can see on my left-hand side. I'm still struggling with a hiccup. This so-called, they call it akasekere, but which has been very painful. So you will not see me in the second video, but you will be seeing my materials. Thank you very much.